Hello and welcome to Across the Life Course. I am Dr. Eunice Lobo, and it is a pleasure to welcome you to the very first episode of this podcast series from the PHFI Center for Developmental and Life Course Research. This podcast was created as a space for conversations around how health and human development are shaped across the lifespan, from early life and childhood through adulthood and aging. Through this series, we hope to bring together researchers, clinicians, educators, policymakers to discuss how life force science can inform research, practice, and policy. Today's episode is especially meaningful for us as we celebrate the completion of the first year of the center. Over the past year, the center has worked to foster interdisciplinary research, collaboration, and dialogue in developmental and life force science. And we felt there could be no better way to mark this milestone than by beginning the series with two distinguished scholars whose work has significantly shaped the field globally. We're also pleased to share that both our speakers today are members of the advisory committee of PHFI CDLR. It is my great privilege to welcome Professor George Davey Smith from the University of Bristol. Professor George is a clinical epidemiologist whose research has pioneered understanding of the causes and alleviation of health inequalities, life force epidemiology, systematic reviewing of evidence, effectiveness of healthcare and health policy interventions, and population health contributions of the new genetics. His major contributions include demonstrating the influence of childhood socioeconomic deprivation on adult morbidity and mortality, methodological developments in meta-analysis, elucidating socioeconomic differences in morbidity and mortality across countries, including the UK, the US, Norway, and India, and the development or an application of the Mendelian randomization approaches to understanding causal relationships in health research. His work has also contributed to the application of causal analysis approaches, epigenetic data. Professor George is an ISI highly cited researcher, fellow of the Royal Society, foreign member of the Royal Netherlands Academy of Arts and Sciences, foreign associate of the National Academy of Medicine, and the fellow of the Royal Society of Edinburgh. He is scientific director of the A1 longitudinal study of parents and children and became director of the MRC Center for Causal Analyses in Translational Epidemiology in 2007, which became the MRC Integrated Epidemiology Unit in 2013 at Bristol University. In November 2025, he was also awarded the prestigious MRC Millennium Medal. So congratulations on that as well, Professor. We are equally honored to have Professor Giridhara R. Babu from Qatar University joining us today as host and discussant. Professor Babu, or as I refer to him as a sir, he's a professor of population medicine at Qatar University. He was earlier professor and head of Lifecourse epidemiology at the Indian Institute of Public Health, Bengaluru, PHFI. He is an internationally recognized epidemiologist whose work aims to reduce the burden of infectious diseases and to address the developmental and environmental origins of infectious and non-communicable diseases in low and middle income countries. Professor Babu has more than 20 years of experience across South Asia and the Middle East with extensive contributions to infectious diseases epidemiology. He established a life course epidemiology program at the Indian Institute of Public Health, Bengaluru, which is now the PHFI Center for Developmental and Life Course Research. So thank you so much for that, sir. Ranked among the top 2 scientists globally by Scopus Clarivate, Professor Babu has authored over 200 peer-reviewed publications, mentored more than a dozen doctoral students. He has also established the Mastery Birth cohort in Bengaluru through his fellowship via the DBT Wellcome Trust India Alliance. He also holds visiting professorships at Maastricht University in the Netherlands and Public Health Foundation of India. With that, we are delighted to begin the inaugural conversation. So I invite you, sir, to take the discussion forward.
I'm extremely grateful to you for agreeing and giving this time to discuss with us. This is my first podcast as a host, so I'll improvise probably while we get started. So, Professor Jad, before we move on to the science, I just wanted to get your perspectives as a person first. And you came to epidemiology from clinical medicine. what pulled you towards thinking about the populations and how these ideas shaped your own career so far?
Yeah, thanks for the firstly, thanks for the invite to speak to this podcast, and then for the question. Yeah, so I'd sort of, as a medical student, I got interested in research. So, and I did an intercalated degree in psychology and a 1 year research project in that. Um, so I at one stage I thought I might try and do a PhD without actually finishing my hospital doing my hospital placements that you in the UK you do for qualification. So when I actually went to do my hospital placements, I'd already sort of decided that I was interested in research. So I mean, I enjoyed junior hospital jobs, but partly enjoyed them because I knew they were going to stop. So and that I was going to move on to something that I liked, and I just had a passion for research, and in particular for population studies. And I was lucky enough to get post after my hospital jobs to actually get a post in Wales, linked to the Medical Research Council epidemiology unit, South Wales, which was being directed by the wonderful Peter Elwood, who is still going strong at 95, and was the man who really pushed aspirin and set up the trials, the first trials showing benefit of aspirin in cardiovascular disease secondary prevention, amongst many other things that he did. And Archie Cochrane of the Cochrane collaboration, who had been the inaugural director of that unit, would be upstairs in the top room talking to people and doing the Times crosswords. So that was I was very fortunate to interact with such people in epidemiology from the start.
That's really interesting. So that brings the question towards the context and the settings. So you mentioned that the ecosystem was powering you to pursue research. And at that time, Glasgow and Bristol environments where you grew up had a strong focus on social medicine tradition, be it Jerry Morris, Jeffrey Rose. How did this landscape feel from within you as a researcher, and
Yeah.
how much of that influenced your work so far?
Yeah, so Jerry Morris was my supervisor for my doctorate, my MD, which was based on the first survey I did in Wales, which was a physical fitness and other factors. So, and of course, Jerry was a person who had been very heavily involved in inequalities in health work. His first paper being in the late 1930s. And he'd been his two passions were social inequality and reducing social inequality, and he became the director of the MRC Social Medicine Unit after the Second World War, and his second was for physical activity and research in physical activity, which I was involved with, and he became a very close friend and mentor, and my first sort of lectureship job was at the London School of Hygiene, working for Jeffrey Rose. So I worked directly with these people. So again, it was a remarkable privilege to work in that context. But that meant that I through Jerry in particular, I had a considerable interest in the history of social medicine, and the approaches of social medicine which really developed around the period just before, during, and after the Second World War. where the notion was that there was major social influences on health, and that you couldn't treat the individual as the only unit that was of interest in epidemiology. And that's something that's definitely remained with me, and indeed on my current areas of interest, thinking about exposures which are sort of ubiquitous within geographical regions. That was very much the notion of social medicine, was you had to think about social distribution as well as individual levels of risk. And of course, with Jerry being the person who really developed academic social medicine in the UK, the person who had the longest career in that and the most influential. Jeffrey Rose was the person who codified the notion of sick individuals and sick populations. And if anyone listening to this podcast hasn't read his 1985 paper, it's only 7 sides long, sick individuals and sick populations. That is, I think, the one thing I would actually say is a central reading for any epidemiologist is that paper. And I go back to it again and again and read it, and I always reread it, and I always find new things in it, that it was it's incredibly concise and just packed full of ideas.
That was actually part of my question. So regarding this essay, does that framing still hold up, or has the field moved past it?
No, I think absolutely it holds up, and I think one of the issues with much contemporary epidemiology is that it's just based upon these very huge cohorts, which are amazingly powerful and wonderful resources, but cohorts like UK Biobank, 500,000 people, of which I'm one, have things measured, and we've all been sequenced, genome sequence and metabolomics and proteomics, and very detailed phenotyping, although not really that many sort of follow-ups. It's basically, but we're middle-aged and older, and that first thing that means you focus on the middle-aged and older. And I think one thing that's increasingly clear, and of course is important to a group interested in Lifecourse epidemiology, is that many of the things in disease, development of maintenance of health, and development of illness happened before then. So those cohort studies make us focus just on one period in life, and secondly, they make us look at the individual level risk rather than thinking about the context of the risk of the individuals. So I think Jeffrey's paper is, I think it's at least as relevant as when he wrote it, that we need to maintain a view of collective influences rather than just individual influences on health. And one thing with etiological epidemiology, with epidemiology searching for causes of disease, it's not. It hasn't developed maybe as quickly as some people thought it would in terms of identifying the causes of diseases which are clearly, in principle, preventable. I mean, if you think in terms of cancers, that's very clearly the case. We don't know the causes of in any coherent or definite way of pancreatic cancer. We don't know how we should prevent pancreatic cancer, for example. We don't know how to prevent colon cancer. And in fact, Richard Doll, another one of the sort of British founders of post-Second World War non-communicable disease epidemiology, as it was called, in 1977, Richard Doll reviewed work on about geographical differences in disease, what happened to migrants when they moved from one population to another, what about time trends of disease, what diseases which are either increasing or decreasing, all of which tell you that in principle the disease must be preventable. and a large proportion of the disease burden must be preventable. And he wrote that he saw no reason in 1977 why the main causes of all the common cancers wouldn't be discovered in the next 10 to 15 years. And of course, that didn't happen. I mean, smoke. He'd of course been very important in the identification of smoking as the cause of various diseases and various conditions. And I think the notion was, or it was absolutely reasonable to think that those sorts of methods which identified smoking as a cause would identify the modifiable causes which must exist for these other cancers and other conditions. But he wrote about 15 years after that. He wrote that he mentioned his prediction and said, but this hasn't happened, even though we'd learned, as he said, we'd learned vastly more about the cellular biology of cancer. We hadn't learned very much more at all about prevention. And in truth, the major factors that were identified weren't what people thought they were going to find. They were infections, human papillomavirus for cervical cancer, H. pylori for stomach cancer. So the expectations weren't delivered.
Yeah, is that partly because much of the focus of the industry has been on finding molecules that will give them probably a blockbuster income, or is it because the research geared towards prevention is not funded well?
I think both of those have played some role. I think an additional thing though is that maybe we're not looking in the right place. We're looking in middle-aged people, as I said, for the factors which cause their disease, and what H. pylori and HPV tell us is that, in fact, the acquisition is often in the case of H. pylori, in infancy and early childhood, and with HPV in adolescence, very early adulthood. That's when the actual the population level driver of the conditions occurs. So we're looking in the wrong place with respect to age. And maybe we're also looking in the wrong place with respect to factors that we think will vary within relatively homogeneous places. Relatively homogeneous countries like Britain, where the exposures might be important. Exposures might be essentially ubiquitous within different places, and so the old forms of ecological study, looking at how exposures differed between places, still has a place, in my view. And this is being shown up by some of the latest technologies and the latest approaches. For example, the study on cancer of the kidney, parenchymal kidney cancer, which we're using sequencing of the tumor. The sequencing of tumors have a large number of signatures which relate to exposures, some of which are known, some of which the exposures are unknown. And in a recent Nature paper, what was found was that in Japan, for example, which has had a substantial rise in kidney cancer, 70% of the cancers in Japan contain a signature which is virtually unknown anywhere else, which suggests that there's an exposure which is only basically common, maybe even ubiquitous in Japan. So I think maybe we're looking at in some cases we might be looking at the wrong place. as I say, when looking at individual level data, that wouldn't work if the exposure is ubiquitous. And in the Jeffrey Rose paper I mentioned, he introduced a famous thought experiment, which was imagining that every single person, if in a place, in an area, smoked exactly 20 cigarettes a day. If that was the case, within that population, you couldn't identify smoking as the cause of lung cancer. That was a thought experiment that stuck with me for many years, and I'm increasingly interested in infection, and in particular in infections in which the age at which people acquire the infection has a large effect on the outcome of the infection. So if you take Epstein-Barr virus infection, across the whole world, virtually everyone across the whole world will eventually become infected with Epstein-Barr virus. And once infected, you retain the viral genome in your B cells, in a small proportion of your B cells for life. Yet we know that if in a rich country, if someone is infected with EBV when an infant or young child, then often there's no consequence. There's no obvious health consequence of that. If you're not infected until you're an adolescent or young adult, then infectious mononucleosis or glandular fever, which is a sort of post-viral fatigue condition, then the risk of that increases very substantially. And for the small proportion of people who don't get infected till their mid-20s, say, they then have about a 30-fold risk of multiple sclerosis. So you're looking at something where the age modifies the outcome of the infection dramatically. But if you just measure antibodies in middle-aged people, then everyone's got evidence that they have been infected with EBV. So you're going to totally miss this important cause.
Yeah, that brings to a related epi question in terms of, you mentioned age modifies the effect of the infection, right? So as an effect measure modifier, be it age or gender or others, have they been studied really well, both with respect to infections and other modifiable factors? What is your take on this?
No, I think they've very often not been adequately investigated, and that there can be combinations of exposures required to generate disease. So, to stay with the Epstein-Barr virus example, in rural Africa, then infection is very early and often appears with a high infectious load. And if people are infected early with a high viral load, and they live in a holoendemic malarial region, where they will certainly become infected with malaria repeatedly, then one sees the high risk of Burkitt's lymphoma, which is the commonest childhood cancer in Africa, but is largely restricted to the regions where malaria is holoendemic. So, and that's I'm very interested in a method called Mendelian randomization, where you use germline genetic variants to help understand the causes, the modifiable causes of disease. And in that case, of course, you have strong genetic effect modifiers of the risk of malaria in sickle cell condition. And people who are heterozygous for sickle cell have a lower risk of Burkitt's lymphoma, because they are less likely to get more severe malarial infection. So, I mean, that's one that I think a very elegant way that a sort of cause has been decomposed. And what one wonders if Burkitt's lymphoma was or a disease of similar um, of similar severity and importance was happening in wealthy countries. There might have been rather more effort on developing EBV vaccines, for example.
So that brings to my next question in line on Mendelian randomization, because you mentioned about it. And for most people who don't know Mendelian randomization, obviously they should go back to your first ever paper on Mendelian randomization, with which we all got educated on how to do a Mendelian randomization study. Since then, the field has sort of witnessed many studies, and then we see you recently very vocal about the misuse of Mendelian randomization as such. So, for a prospective AP student, what would you advise, starting from the basics till now, and then what should be the way they use this tool or this design for their research?
Yeah. So the sort of trick in Mendelian randomization is that you use a genetic variant or multiple genetic variants, which can be taken to proxy for an exposure. So if you measure the exposure itself directly, which would seem the obvious thing to do, then you have the problems of confounding, whereby factors which also influence the outcome of the outcome that you're interested in influence the exposure level. And a very important subcategory of confounding, which is the reverse causation, when the disease process influences your exposure. So if you consider something like atherosclerosis, which develops from late adolescence on, atherosclerosis is an inflammatory condition, and atherosclerosis increases your risk of a heart attack. But because it's an inflammatory condition, it also influences many things like cytokines in particular, and acute phase reactant proteins like C-reactive protein. So you get strong and highly consistent prediction that higher CRP levels, predict coronary heart disease, suggest a higher risk of developing coronary heart disease, but it's not a cause. And one thing which helps demonstrate it's not a cause is that there are variants in the CRP gene itself, which relate to greater gene expression and higher CRP levels on average. So the people who carry one set of variants will have higher CRP levels as a group. Once you treat them as a group, not as individuals, you group them according to their genetic variants that they carry that relate to CRP levels. They would have a higher risk of coronary heart disease if C-reactive protein was a cause of coronary heart disease. And you have positive controls, like people who have variants which mean they have higher LDL cholesterol levels, higher ApoB carrying lipid lipoproteins, and they have higher risks of coronary heart disease. So you see both these positives, where something has been shown to be a cause by treatment in trials, and you also see nulls, where you see things which were being considered to be important factors, like C-reactive protein and HDL cholesterol, which was even called good cholesterol in popular terminology. But when the randomized trials were done of using drugs that worked through a whole variety of mechanisms, when randomized trials were done which increased people's HDL cholesterol, they showed no effect on coronary heart disease outcomes. So HDL cholesterol appears not to be able to protect against coronary heart disease. And that's one of the examples where Mendelian randomization has been valuable, is showing things which actually aren't actually going to be good targets for either pharmaceutical or lifestyle manipulation to try and change them to reduce disease risk. So So the key assumption for Mendelian randomization, or the principle for Mendelian randomization, for me, is that of gene environment equivalence. And by that, we mean that if the exposure of interest is at a different level at a group, between groups, if it's at a different level because of the genetic makeup, because of the genetic variants that people carry versus the sort of counterfactual different set of variants that they could have. If it's that counterfactual difference in genetic makeup that influences or the exposure, it will have the same downstream effects, the same downstream consequences on disease as if you're manipulating the exposure by certain forms of plausible manipulation, be it pharmaceutical, be it lifestyle, etc. So to carry out a worthwhile Mendelian randomization study, you've got to ask yourself the question of whether gene environment equivalence is at all plausible. And if gene environment equivalence is not plausible, then you should stop. And in many cases where Mendelian randomization studies are now being done, it's very clear that gene environment equivalence is just simply not plausible. I mean, you see people, because everything is heritable, if you do a genome-wide association study of particular habits, like whether people eat breakfast regularly, you do find genetic variants that relate to eating breakfast regularly, but they are not doing so through any proximal relationship between them and eating breakfast. They're doing so through upstream processes, like risk-taking behavior, or maybe smoking behavior, or a chronotype, your tendency to be a morning person or evening person, or educational level. They're doing so through these other factors. They are not in any way direct influences on the behavior, like breakfast skipping. But the problem is that because Mendelian randomization is largely done these days in a two-sample setting, all you need are summary data from genome-wide association studies for both your exposure and your outcome, and you can calculate some relative risk or whatever effect measure you want. So you can do it, and sadly you can publish papers, but they're not meaningful because the basic principle, that of gene environment equivalence, doesn't apply. So there should be less Mendelian randomization papers published, but there should be better. There should be better papers, and also I think I'm an advocate of a triangulation of evidence perspective, whereby for any question where you want to improve causal inference, you think of a series of different approaches which could get you evidence as to whether that is a likely cause, and address the question through a series from a series of different angles, applying these different approaches. And then when the different approaches agree, then you can have a stronger sense about whether things are causal or not.
Yeah, that's a great primer for most people who want to know about Mendelian randomization. I would recommend definitely to read into the details of this. But I just also wanted to note, very often people who invent a new technique or a new tool are the ones who will propagate it. But here is one instance where the person who originated the idea is calling for uh, a lower number of papers, mainly because of the improvement in quality. So this is a rare thing, and I definitely compliment you for educating and continuing the advocacy. But on that note, to improve the causal inference, you mentioned triangulating the data and then making sure that we get the proper research question addressed. Recently, emulation of target trials has been sort of in the attention for improving causal inference. What is your recommendation on how do we approach?
I mean, I think just like Mendelian randomization depends upon the sort of this fundamental principle of gene environment equivalence. I think that with target trials, they can both produce useful findings and produce things which are clearly which are erroneous. So it depends upon the assumptions, and one needs to work through the assumptions that are required. And essentially, target trials do not account for measurement error or unmeasured confounders.
Yes.
So the advantage of one of the strengths of Mendelian randomization, when it is correctly done, as it is, as in situations of a protein, for example, or a very obvious drug target for which there are genetic proxies, is that it is a method which can help alleviate or avoid measurement error and unmeasured confounders. When done correctly, there should not be unmeasured confounders. With target trials, then that isn't the case. You need to think about is it really plausible? Is it plausible that there's no important unmeasured confounders and measurement error in the confounders? What target trials do well and unequivocally do better than some of the naive observational analyses of interventions is target trials deal with selection biases, which can be really important in getting the wrong answer in observational data. So you've got to think about whether you're looking at a situation where the selection bias is occurring and is generating the difficulties of interpretation, rather than ones when it is unmeasured confounders and measurement error. And I think people interested in this area should look to find examples of when the target trials have been carried out before there were actually any randomized controlled trials of the issue. I mean, that's the situation which is obviously the most challenging and the one which would actually increase confidence in the approach. And there's not many examples that I know of that. So I'd say look around at what's been done and make your mind up about whether the situation you want to study fits with the approach, and whether and think about study situations where people have done target trials before randomized trials have been published.
Absolutely, that's very useful insight, Professor Smith. That brings to another related question on when researchers pursue either grant seeking or their own research interest. Carrots prevented cancer, therefore the alpha tocopherol and beta carotene became the trials, and then we know how things went wrong afterwards. Still, carrots prevent cancer, but neither the focus on improving carrot intake have taken the importance of public health interventions, mainly because the pharma did not get the beta carotene as supplements to do with. So what is your advice to the young researchers on how do they set this agenda? When do they know that they're wrong, and how do they do course corrections? What's your advice based on several successful research initiatives you have done? Also, many that you, in your MRC address, you said the ones that did not get funded are the ones I was probably not very happy doing it. So I'm happy that they didn't get funded. So I just want you to reflect on that.
Yeah, well, that was, of course, in retrospect. I didn't have the time. I was very cross, but I've been lucky. I've certainly been lucky with things that did get funded, and I've been lucky on some occasions with applications that I put in that I'm glad they didn't get funded, because they would have
been complex.
spent a lot of time to produce null results. So but I just to revisit one thing you said is that there are obviously examples in nutritional epidemiology, many examples of when observational studies suggested something was going to be strongly protective. For example, use of vitamin E supplements. So these were a couple of very large studies from the US, which found that people who used vitamin E supplements had a 35% or more lower coronary heart disease risk than people who didn't. And they did all the usual things of measuring all the confounders they could think of and adjusting for them. And the statistical adjustment had rather little effect on attenuation. But then when the trials were done, precisely the same thing as was being looked at in the observational studies of actually randomizing people to a large range actually of different formulations of vitamin E versus placebo, that no benefit whatsoever was seen. And this, I think, is the issue that I think is underappreciated, even though all people who've been who have done epidemiology courses and training will have heard about it. Maybe they think too much, which are the issues of measurement error and missing confounders. And you can show that because of the level of error that exists in single or even a couple of measurements of a factor, like some dietary intake factor or some potential confounder like smoking, social position, and health-seeking behavior, and so many such factors. These things we're not measuring very well. And if you do simulations, you can show that given the level of measurement error that we know exists in factors where we've actually done repeat studies to see how things change over time, and how much measurement error there is, that measurement error will often lead to the sorts of findings you see, that even something where the truth is null, there's no influence of the factor, causal influence of the factor on disease, you would see a risk, a level, relative risk, and then you would adjust for the confounders and it reduces by about 30%. That's actually what you should expect for something which is actually null. And that's what's been seen in many of these cases of things which have been shown later in trials to be null, is that the adjustments, only attenuate the associations to a small degree. So I think there should be sort of considerable skepticism about statistical adjustment as a way of making decisions about causality.
Yeah, I think that's the position where, since we both are advisors on Center for Lifecourse Research, also looking at development through the Lifecourse, that brings to the point of what is our personal belief on what things can work versus what cannot work. And that should be the genesis or sort of an idea to discuss about Lifecourse epidemiology. So you have witnessed different phases of Lifecourse research being done through the entire globe. What do you think, according to the strength of the Lifecourse AP as a discipline? What is it solidly proven? Where is it overextended? Or how is this Lifecourse framework from a research perspective that will define the future epidemiology pursuit?
So I think an example where there's very clear differences in outcomes according to stage of life when exposed I think probably the best example is Epstein-Barr virus infection. I mean, but infection hasn't been much interest in much life course epidemiology. I mean, life course epidemiology was certainly stimulated by the David Barker's fetal origins of health and disease concept, which I thought was enormously attractive and sort of made sense in terms of fetal development as being an important period within the life course. But I think much evidence has shown that it does not have the very large sort of effects that were thought it may have. And this includes, for example, Mendelian randomization, using genetic variants which have their effects during fetal development. Now, one of the pin-up examples in our first paper on Mendelian randomization was looking at maternal folate levels and neural tube defects in the offspring.
And using MTFHR.
Yes, using the maternal carriage of -677 PHFI variant, the TT, or the sort of oversimplifying for want of time, but which was like a mimic of a sort of low folate state, the TT genotype. And if the mother carried the TT genotype, then her offspring had twofold risk of neural tube defect. And you had a beautiful negative control because, of course, you had the father's genotype, and the father's genotype had no impact on the neural tube defect risk in the offspring. And it's a beautiful negative control because any confounding would apply equally to the fathers as to the mothers. And then, of course, randomized controlled trials showed that folate supplementation reduced the risk of neural tube defect. So that's a case where there definitely is a fetal origin of a serious health outcome, which is neural tube defects, and clearly there could be and are likely to be fetal origins of other things. But genetic variants looking at maternal adiposity, for example, have shown, contrary to what is reasonable to expect, that has small, if any, impacts on cardiometabolic and other outcomes in the offspring. And maybe biologically one thinks that the fetus is a very particularly protected stage of life because the mother is buffering the fetus against external shocks and provides that buffering function. Maybe we're focusing too much on antenatal life and not enough on the relatively early postnatal life, including infections during those periods. But infections are very difficult to study. I mean, there is always the cliched story of the drunk man looking for his keys that he's dropped, and he looks under the lamppost because, and when asked, he says because that's where the light is, not because that's where he thinks he dropped his keys. I think a lot of fetal origin, a lot of life course work was based around things that one could get measures of retrospectively, like birth weight. Those are the things that we're focused on. And my feeling is we need studies prospectively that collect, for example, infection data, frequently over and repeatedly to identify when people first become exposed to infections, responses to them, etc. Now that's a very expensive and time-consuming process, but unless we start doing studies like that, we're not going to find out the answers to those important questions.
That's a great insight, and definitely the audience will love the example you gave, and then they'll go beyond the lamppost to find where the keys are. But on that context, the classical theoretical shift between critical periods of exposure versus accumulation of risk. From what you stated now, does that still hold good in terms of this distinction, or have they collapsed in practice? At least from, let's say, building new cohorts.
Yeah, so I think that it's not going to prove, it's unlikely to turn out that it's sort of one model applies to all conditions. I mean, I think there are definitely sensitive periods in relation to infection, for example, and probably in relation to some nutritional shocks and and other insults. And for some things, maybe things look more like they're more straightforward accumulation of exposure. I mean, the example which has always been given as one where, at least observationally, that's what it looks like, although there's no great experimental data, would be like lung function, for example, and respiratory disease, where it does, there do appear to be early life influences, but then smoking and occupational exposures and etc. In later life clearly have very strong adverse consequences. So I don't think it's going to be a one size fits all model at all. I guess that's one thing where I which is something I have written quite a lot about, which is suggesting that there isn't this general susceptibility. There aren't these sort of general processes that influence many diseases. I think we have to think of condition specific risk rather than general processes.
Looking at the data, whether it's Danish national birth cohort or other Scandinavian data sets, would you get still information on infections at different stages of life? Is that possible?
No. So this is the issue: is that unless you collect blood samples, for example, repeatedly, then you can't. We have no way currently. I mean, it'd be fantastic if some ways were developed, but we have no way currently of actually identifying the age at which an individual got infected with an agent, from measurements taken 20 years later. So this requires actually establishing new cohorts, which I think we should be doing. I think I think we should be establishing cohorts that start doing this, especially as the technologies and cost of assessing a vast number of antibodies is going down, and the technologies of collecting blood samples is improving. I think we should be establishing those cohorts now, and if we don't do so, we'll be in the same position in 30 years' time without having the necessary infrastructure to be able to examine these things.
Hopefully, some of the funders are listening to this podcast, and for them, if they want to learn from this podcast, what would be an ideal cohort that you would want to build? What should be the infrastructure?
So I think, the birth cohorts that recruit, women when pregnant, or even even more expensive and complicated studies that recruit before conception. I mean, I think those cohorts that then have take repeat blood samples on the children after birth, through to adulthood. That's what we need is cohorts like that, and we don't need hundreds of thousands of them. We just need a few that are reasonably representative of the global disease ecology to be built up. That would allow us to actually look at these issues, and we should be starting now. I hope the fund. I very much sadly I doubt any funders will be listening, and I doubt they would be swayed by my arguments. But that's my view.
But Professor Judge, I can reflect here. When we wanted to start the cohort, I was told nobody will fund the cohorts. So we kept going, and then eventually we got funded. I hope I'm optimist there. Probably somebody will listen to. But so just for them, one is the blood infrastructure. Would you also need microbiome, saliva, or placenta?
Yeah.
What kind of interest?
Yeah. And again, no one will know what. No one knows at this stage which will prove to be more valuable. I mean, my perspective, I would start with blood. I think collecting saliva and even swabs, if there's a way of doing that, would also be worthwhile. I think, and and it might prove to be the case that the microbiome. I mean, there's so many different microbiomes. There's the gut microbiome, the vaginal microbiome, the nasal microbiome. There's many different microbiomes. But I would, if one was investing in a cohort and had limited investment, then the repeat blood samples is what I would invest in.
Since we are coming towards the end of our allotted time, I am conscious of this. So I have two very important questions before we can end. For a young researcher in low and middle income countries drawn to this kind of work in epidemiology, what would you tell them where to put their effort and what they should not ignore? And if you were to restart your career now, that will be the counterfactual of how you can explain.
Yeah, so I'm absolutely loath to give advice on what people should do, because I think sadly prediction is very hard, particularly about the future, as I think Yogi Berra was said to have remarked. But I can say what I would do is I would have developed an interest in infection at an early age. I mean, actually, I did work including in India and elsewhere. I did work on HIV and childhood diarrhea to a smaller extent, but I would have had a more generic interest in infection. And I think one thing, if looking back at the sort of history of what became known as non-communicable disease epidemiology, epidemiology was infection, and in the first epidemiology textbooks that were written were simply textbooks of infectious disease. I mean, Major Greenwood, in his book about crowd diseases, which is one of the early texts about epidemiology, did actually, in 1936, did actually have a chapter on cancer, I think it was. But the vast majority was infectious disease, or it was all infectious disease, and it was only Jerry Morris's Uses of Epidemiology was the first, textbook of non-communicable disease epidemiology in 1957. And there were all these famous quotes. The Surgeon General in the US in the 1970s saying we were closing the book on infection, and that was definitely the environment that I started in epidemiology. Everyone was looking at diabetes, coronary heart disease, peptic ulcer, that had just started to fall, and cancers, thinking that they would prove to be that they were diseases of the modern environment, and of course peptic ulcer turned out to be H. pylori, driven by the population level, driven by H. pylori, and H. pylori eradication improves the condition, and stomach cancer similarly. So rather ironically, quite a few of the diseases that the so-called non-communicable disease epidemiology focused on turned out to be to have an infectious origin. I mean, if you look at the greats post Second World War epidemiology, Mervyn Susser and Zena Stein, for example, who did amazing work on peptic ulcer, still thought it was created by the modern environment. Jerry Morris and Richard Titmuss did amazing work on peptic ulcer. But in his book, in the third edition of Uses of Epidemiology in 1975, Jerry said, he reflected on the fact that peptic ulcer rates had gone up and down in a birth cohort fashion. He said, the theories that most people had, that it was like it was a stress-related disease. He said it's just not imaginable that stress in modern, in Britain, where he was looking at data, had sort of gone up and then come down. It just wasn't what happened. So, and that's a sort of triangulation viewpoint of saying, you need to look at disease levels ecologically over time, between places and between groups within a society, etc., to actually understand causes. And if things don't fit, then you should question your models.
Yeah, I think that would motivate at least a fraction of the listeners to become epidemiologists if they not already are. But once they decide, the biggest challenge, and this is my last question, is that you've been such a great mentor, and we have had several students of yours, who, including Dr. Kindra, who was our long-standing collaborator. That kind of mentoring system doesn't exist in the global south, or it's just evolving. What is your advice on developing these mentoring frameworks, and what is the way in which the institutions in the global south can focus on it?
No, I think that's a brilliant and a really important point. I'd say, I mentioned, the people who I was lucky enough to work with, learn from. Also, people who've been incredibly influential were people who were students with me. When I was doing the master's course at the London School of Hygiene and Tropical Medicine. People who've stayed friends and colleagues for years, and so I think you should think of horizontal mentorship as well as vertical. I mean, you should. I think building strong networks of people who are sort of co-learning together, and and part of that is actually challenging the ones. I won't go into any particular cases, but I certainly would occasionally challenge the views of the people who are employing me or teaching me or whatever. The International Epidemiological Association, for example, I think could take a view of trying to actually help develop mentorship schemes. I mean, I've been involved in a few, but they're UK based, but a few schemes where and they're schemes whereby the mentees choose a mentor. You get people who say, yes, I'd be happy to do this, but they don't select the people who they interact with, but they just say that my name can be in a list of someone who people can choose because they're interested in similar things. And I think if you try and the few situations I've seen where those have been actually well organized, they've worked really well. And I think it's worth trying to think about developing that sort of model where it's a mentee chooses model. And I think you would find there'd be quite large numbers of a lot of people, including you don't want to get grand old men. You certainly don't want old men. So I have to excuse myself from contributing to such a scheme anymore. That's the last thing you want. You want diversity. You want diversity of age. I mean, I think the people who can be the most valuable mentors are mid-career, people in earlier career stages, when they're still flexible in terms of how they think, and would also benefit enormously from interacting with mentees. So I do think that those schemes could be built. As I say, I've seen a few schemes that work well, but I think thinking about how to do that would be an important thing.
Mr. Smith, thank you so much for your time and joining us. This will definitely influence and inspire a great generation of epidemiologists in India. And we'll continue to learn from you as the advisor on the CDLR at PHFI. Thanks so much for your time. And I really enjoyed listening to your life course perspectives on life spent in epidemiology. Thank you so much.
Right. Now, thanks very much for asking me. It was an honor to do this. Thank you.
Thank you, Professor Smith. I'm really sorry we might have overshot your time commitment session. Thank you so much.
Fine, yeah. Lovely. Take care.
Take care.
Bye. Thanks, everyone. Bye.